Hello, and welcome to the Oxford Centre for Personalized Medicine's lecture by Professor Heidi Rehm, in honor of the memory of Dr. Stanley Ho. I am Ian Hewen, a trustee of the Dr. Stanley Ho Medical Development Foundation and member of the CPM's External Advisory Board. The foundation aims to enhance the quality of medical service and delivery in Macau, Hong Kong, and other regions of China, and enters into academic partnerships with relevant worldwide organizations to achieve this. It is our pleasure to have supported the Oxford Centre for Personalized Medicine since 2013. The University of Oxford's Centre for Personalized Medicine is based at St. Anne's College and the Wellcome Centre for Human Genetics, United Kingdom. Its principal aim is to engage a wide range of interested people, including academics, clinicians, students, patient groups, policymakers, and the public with personalized medicine. Dr. Ho's support for the center marks his vision and commitment to help transfer knowledge of personalized medicine to healthcare professionals and students worldwide. Dr. Stanley Ho passed away in Hong Kong at the age of 98 in May 2020. He was not only an extraordinary leader, but also a great philanthropist. His legacy is his extensive altruistic efforts. The foundation will honor Dr. Ho's memory by continuing his spirit of serving our communities. This lecture forms part of a series of talks commemorating 100 years since Dr. Ho's birth. It gives me great pleasure to introduce tonight's speaker, Professor Heidi Rehm. She is co-director of the program in Medical and Population Genetics at the Broad Institute and the Chief Genomics Officer in the Department of Medicine at Massachusetts General Hospital. Dr. Rehm is an outstanding genomic medicine researcher. Her work as founding director of the Laboratory of Molecular Medicine and her leadership of the Center for Mendelian Genomics at the Broad Institute have resulted in a genetic diagnosis for thousands of patients and the discovery of hundreds of new disease genes. She is also a pioneer of open data sharing for clinical genomics. She co-leads the ClinGen project, is vice chair of the Global Alliance for Genomics and Health, and was instrumental in the development of the data sharing platforms Clinvar and Matchmaker Exchange. The impact of Professor Rehm's work in this field cannot be understated, and we are delighted to have her with us this evening. She joins us now in the early morning in the United States in order to speak to audiences across the globe, including at lunchtime in the UK and the evening in Hong Kong. Her title is Creating a Global Ecosystem to Support Genomic Medicine. Professor Rehm, it is my delight to welcome you as the second Oxford CPM's Dr. Stanley Ho Memorial Lecturer.
Thank you. Thank you very much for that wonderful introduction. I'm just delighted to be able to give the second Stanley Ho Memorial Lecture. I'm going to share my screen now so we can see slides. A second. There we go. So welcome everyone, whatever time zone you're in. As was mentioned, I'm going to talk about how we advance genomic medicine through global collaboration and data sharing. One of the things I thought would be fun to contextualize the work that I'll talk about today is to put it in the context of 10 bold predictions that were made by NHGRI, or the National Human Genome Research Institute, part of NIH in the US, and through an effort of defining their strategic roadmap for the next 10 years, they also defined 10 bold predictions. And they did this in collaboration with many leaders in the field, trying to come up with, what could be bold, but really trying to be achievable goals. I won't have time to go through all 10 of them. You can go to the NHGRI website and hear talks on these and more details, but I'm going to start with the second one. The biological function of every human gene will be known. For non-coding elements in the human genome, such knowledge will be the rule rather than the exception. Now, to make this a little more fun, I thought we'd actually do a little audience poll after each of the 4 of these predictions and get a sense of where you are as an audience on these bold predictions. So somebody's going to paste into the chat the URL, but it's also just right at the top. You can just go to any computer and type in pollev.com/Heidi Rehm 026. And then you'll be given the option to respond, or you can use your smartphone and text to 22333, the words Heidi Rehm 026, and then that'll allow you to then put in A, B, or C and respond as such. So it looks like it must be working because there's at least some responses coming in. So I thought this would be fun. So we're about 50-50 on the we'll get close but not at all, or I'm skeptical. Actually, B is now starting to win out here. So, all right, we're starting to level out with enough responses to be stable. So it sounds like many of you think we will get close, but not probably quite achieve it. A few others are a little more skeptical. So let's now talk about how we're actually going to get there, and some of the work that we've been up to try to do that. So this is our Broad Center for Mendelian Genomics, and also we run a project called Rare Genomes Project, and we're now part of the Gregor Consortium, which I'll talk about in a second. So just a wonderful team here led by Anne O'Donnell-Luria, myself, and Mike Talkowski, and we appreciate the funding from NHGRI, the Chan Zuckerberg Initiative, and Illumina. And our work has been focused on really trying to discover novel causes of rare disease. Get my slides to advance. There we go. Now, we've done that successfully largely because we collaborate extensively with key leaders in the rare disease field all around the globe. We've amassed over 15,000 samples from 7,700 families because we've been working with over 45 collaborators from 57 countries, working across 57 countries. And that is how we've been able to be successful, really, as making this a collaborative effort. We focus on many different areas of rare disease, including orphan, very rare diseases. And through those efforts, we've discovered collectively 322 well-supported gene disease relationships, as well as another 604 candidates that still need more case or functional data to support their causality. We also, that's how we sort of work with collaborators to bring in their samples that they've collected around the world. We also have a direct enrollment project called the Rare Genomes Project, where this is restricted to the US because of our consent requirements, but we've been able to enroll hundreds of families around the US and analyze their data. And to date, we've identified 120 candidates, as well as 85 clear results that we've been able to return to families. And all these orange dots are families around the US that we've returned results to. Now, our Center for Mendelian Genomics has been part of an NIH-funded program called the Centers for Mendelian Genomics. And you can see there's 4 different groups here: Johns Hopkins and Baylor College of Medicine, University of Washington, our site at the Broad, and the Yale School of Medicine. And collectively, if we aggregate across these 4 centers, there's been close to 30,000 families that have been analyzed. Lots of data generated shown here, and collectively we've discovered 1,637 what we call tier 1 genes. These are the well-supported gene disease discoveries, and another 2,270 candidates. All these figures and all of this data is in a new paper that's just about to come be published in Genetics and Medicine, probably this week, but it is on a preprint and medRxiv as well. And I'm happy to share these slides publicly. I'll probably post them on the internet if you want these URLs. So to date, we've been able to identify causal gene or candidate for about 38% of families across the entire set of Centers for Mendelian Genomics. But these candidate genes down here, those plague me. Some of these won't turn out to be real gene-disease relationships, but a lot of them are just awaiting some more data. So how do we follow up on candidate genes? And that's where this matchmaker concept that we developed now over 5 years ago is really important. And the idea is you have 2 clinicians or 2 researchers somewhere around the globe. They've sequenced at least one family and identified a candidate. Maybe it's a de novo variant from a trio sequencing effort in a highly constrained gene, but you've just got one case. That's not enough to implicate a gene in disease. But if you find a match, at least one, sometimes multiple around the globe, where at least one of your candidate genes is overlapping and your phenotypic features are overlapping, that can really start to build the evidence for the gene. And we realized that these gene candidates were sitting in people's databases, not seeing the light of day all around the world. But if we just got those to be matchable and brought together, we could really advance and get publications out there and let the field know about these discoveries. So to do that, we developed a platform where we could not make people centralize data, but in fact allow application programming interfaces to communicate. And it's like playing Go Fish. I have, this gene candidate, anyone have a match? And so we have a very small number of fields for this application programming interface that allows you to just submit. And mostly all you need to put in is a gene name. And then if there's any matches, the 2 groups, both the submitter and whoever's data was sitting there waiting to be matched, will both get emails simultaneously. If you include phenotype, a lot of the tools will rank the matches based on how good the phenotype matches. And this platform has been amazingly successful in building evidence for gene disease causality. We've got some statistics on our website, over 40 publications that have been discovered and published genes that had a cross-node matching, as well as over 500 publications just using Gene Matcher. And this is a site that anyone can go and just develop a login and enter your gene. You don't have to have your data stored in any of these nodes. So that site is used by hundreds of different investigators. To date, we have over 10,000 candidate genes across the Matchmaker Exchange, across these nodes, representing data from over 167,000 rare disease patients from over 4,000 contributors from 84 countries. And the submissions from our CMG programs are shown here in the publication that's coming out. So a really powerful platform. Now, sometimes you put a candidate gene into Matchmaker Exchange and you don't get any hits. So we need more people to be putting their cases and their candidates into this platform. In those cases, really working on finding collaborators that can do functional studies on these genes becomes very critical. And sometimes you only have one other match. The disease might not be specific enough for that to be a valid enough match, and therefore functional data also becomes a critical contributor. And so through our Rare Genomes project, we've been reaching out to collaborators around the world who have functional platforms for studying these genes, and by giving them our candidate genes and variants, they're able to introduce them into these assays. So we have 21 functional studies currently in progress from the Rare Genomes Project dataset. Our collaborators, of course, in the CMGs are doing this across lots of gene candidates as well. Publications in progress, really showing the power of collaboration to advance this field more quickly. I do want to just highlight a platform that's been launched at the Baylor College of Medicine as part of the UDN, Undiagnosed Disease Network, where they're really trying to match functional researchers, so we don't have to scour the world. And we don't mostly do that looking for prior publications, but if a group is working on a gene and hasn't yet published on it, you won't find them in the published literature. So this is a way to match researchers doing functional work with candidate genes from investigators. It also connects to an international network, and there are more networks in other countries like Japan, Canada, Europe, Australia, that have their own networks. And some of these networks actually distribute funding when there's been a match between a candidate and a functional group. So really starting to better operationalize a network to advance and scale our rare disease research. Now, I mentioned the Centers for Mendelian Genomics, that program has actually come to an end, but a new program working on rare disease was funded, now called the Gregor Consortium. Some of the sites are the same, shown here, but we have two new sites, Stanford and Children's National, and partnership with Invitae. Similar goals, we're really trying to solve the causes of rare disease, although really trying to innovate on those most challenging cases that no one's been able to solve to date. That will require both innovative approaches as well as really widespread data sharing, where we bring all of these cases together and can collectively analyze them to improve the power. And I will go back to this slide here and say that although we've made great progress in discovering many well-supported gene discoveries and candidates and solved lots of cases, we still have 62% of those cases that remain unsolved and without a gene candidate. And in fact, there's evidence for over 10,000 more genes yet to be discovered from Mendelian disease, from a publication from Mike Bamshad and his group, where they just simply looked at animal models in existence, as well as highly constrained genes from our Nomad database. And that estimate suggested that there's over 10,000 discoveries to be made in rare disease alone. And it is this work that is going to get us to understanding the function of many of the genes in our genome. And I will just highlight a paper from the Orphanet group where they really showed that most of the patients walking around, or individuals walking around the globe with rare disease, actually have that disease due to a smaller number here, 149, of more common rare diseases, when in fact, most of rare disease, over here, over 3,000 different gene disease associations, are actually incredibly rare, present in fewer than 1 in a million individuals. So to tackle this group, which we have largely not tapped into, it will be more challenging. These are very, very rare diseases, and it will be even more critical to share our data globally to find those matches and match functional studies with investigators, with candidates. So we're going to have to do this even better than we're doing it today. All right, so moving on to the second bold prediction. Generating and analyzing a complete human genome sequence will be routine for any research laboratory. So I'm going to ask this question. We'll see whether you're more or less skeptical of this one. So go back to that same site. Looks like some of you are already on it. And we'll capture a little bit of data. See where you all are with this. All right. So, so some of you think this are a little more positive about this prediction actually happening, and that absolutely we will be there and allow every research laboratory to do this. And most of you feel it's pretty much, if you got convinced. And it may be based on how you think about this question, because in fact, for me, it is absolutely how we think about this. And I'll point out that there is constant innovation in sequencing technologies, variant detection methods, and analytical approaches to identify causal variation. So when we talk about routine, what part of it is routine? And are we all using the same, or not the same, but best in class approaches? So I would argue that if we all work together to share tools, methods, and platforms freely, we will all have access to state-of-the-art approaches, and this will be realized, absolutely. It may not be that everybody builds their own pipeline and can do this routinely, having developed the entire end-to-end workflow themselves. I doubt that will allow us all to get there. However, if we all work together, because each group is contributing innovations in these different areas, and if we can all share those innovations into a common pipeline that we can all access, it will be very easy to get there. So I will go back to this figure from the CMGs and point out that all of this data and candidates are now shared on a platform that NHGRI has launched called the Anvil Platform. And this embraces a newer paradigm of how we're working together collaboratively. And that is, instead of putting data into a single centralized database and then allowing people to apply and download that data into their own sites to work with it. This is now a platform where you put the data onto a cloud-based platform, and then people bring methods and tools and analyze the data on that platform. It's actually much more secure in terms of security standards, but it also allows easier access to that data, because these data sets are getting larger and larger, and it's very costly to download them. So all this data is now on Anvil. And in fact, this is a very intricate platform, which a recent publication from the Anvil team is now out, and you can go there and read about it in Cell Genomics. And they have developed user portals for different programs for the tools they need. But anybody, can contribute innovative tools to this platform, workflows. You can create your own storage buckets for your own data, and then allow it to be analyzed in these pipelines. And I think this model is what's going to allow every laboratory to routinely access and contribute to genomic analysis and make it really democratized. And we actually took our Seeker platform, which is a platform to allow individual case level analysis for rare disease cases. We spent a year really making it FSMA compliant, so it could be actually compliant with some of the software tools that are stood up permanently on this platform. You can spin up instances of tools that aren't FSMA compliant individually, but Seeker is now FSMA compliant. It's also open source, so others can actually download the software and set it up on your own sites. You can contribute code to it. Lots of ways that we can, as a community, contribute to this. And because of that new functionality has actually been contributed to different groups that we've been able to put into the Seeker platform, but anyone can load up your data and now use it to analyze freely. So this is a model that I think we should all embrace. Also, all of our data is there. It's not just there, but it's well annotated, so you know which cases have been diagnosed and which are not solved, what the phenotypes are, all of that. And in fact, colleagues are now accessing that data. Aaron Quinlan, this is just an example, was testing out a new structural variant caller his team developed, and they were able to actually identify an upstream non-coding structural variant that allowed us to make a diagnosis in one of our cases that hadn't yet been diagnosed. So just one of many examples of the power of sharing this data so that we can work collectively. Now, I will point out that in order for us to work collaboratively and globally with different methods and data sharing, we have to use better standards so that data and those tools are interoperable. And that brings me to the Global Alliance for Genomics and Health. And I've been part of this just brilliant organization for now over 5 years. We have technical work streams developing the standards across all of these different critical areas in genomics, foundational work streams developing policies and frameworks and regulatory and ethics and data security, critical elements of this. And the way we've been able to get the community to rally together to develop these standards and make sure they're fit for purpose is by defining real world driver projects. And you can see, I'm sure you recognize many of these projects that are well funded and operating around the world. They span rare disease research, complex disease research, cancer, basic biology. And the members of those groups are participating in our work streams, defining the needs that they need to do their work. And then when standards are developing, they're testing them, operationalizing, making sure they work, and iterating on them as needed. This is what's going to allow us all to collectively work around the world. And in fact, this is exactly the approach that allowed us to more effectively launch the Matchmaker Exchange, because we worked with these work streams early on. They had slightly different names at that time, but we worked with what was then the data work group of Global Alliance to develop the standards for the API. We worked with the clinical and phenotype work group to develop how we exchange disease names and phenotypic features. And we worked with the regulatory and ethics work group to define a consent framework for the Matchmaker Exchange, what sharing did require consent and what didn't require consent. Many of the papers of this platform have been published over the years, but it's working with standard organizations that will allow us to innovate and do this work globally. I also will say one of the key aspects of this work that we're going to have to embrace is the notion of federation. And federation addresses many different types of federation. Here in the context of data sharing, you can think about 3 different models of data sharing. One, the traditional model of a central database. Everybody sends their data to one location. And I think that's still a powerful platform. And in fact, probably the best way to approach genomic knowledge bases like, for instance, Clinvar. This is not patient data. It is claims about pathogenicity. So it's much easier to just publicly share. That's sometimes easier to manage just in a single central database. On the other hand, when we're talking about patient data or individual level data, this needs to be often in a secure environment, a secure cloud, when we're talking about large data sets that are costly to manage. And so often within an individual country or organization, they may bring all of the data together. For example, Anvil bringing that data together in the Anvil environment to deal with large-scale data sets. However, if we look globally, many countries can't send their data outside their national borders. And in that case, we will have to deal with federation, where each of those data sets may be separate in other places like the Matchmaker Exchange. And we need to be able to send our analyses to a data set, calculate, answers, and bring back just the answers, which can be shared, the summary statistics, and then aggregate summary statistics together to answer questions. Though that will require even more sophisticated harmonization and interoperability of the approaches we take. So that's what's going to get us there eventually. Now I'm going to move on to number 7. The clinical relevance of all encountered genomic variants will be readily predictable, rendering the diagnostic designation variant of uncertain significance obsolete. So our next poll, do you think this will happen by 2030? Yes, not quite, but we'll make major progress, or we will still have a long way to go. So I was a little more skeptical of this one. This is actually the topic I was asked to speak on for NHGRI's lecture series in these bold predictions. So if you're interested in that specific talk, it's recorded and publicly available. But you're probably with me saying, I think the US will still be around. We'll still have a long way to go. But I also agree with B, which is we will make major progress. But how are we going to make that major progress? So here I'm going to talk a little bit about the ClinGen program, which stands for the Clinical Genome Resource. And this is a large organization, but some of our core activities focus on what we call gene curation and variant curation. Here we're scoring aggregate genetic and experimental evidence in a semi-quantitative approach, and that approach has been published. To define gene-disease relationships into one of these buckets: definitive, strong, moderate, limited, animal model only, no known disease relationship, disputed or refuted claim. And you really have to do that first before you go on to variant classification, because variant classification, you can't call a variant pathogenic for a disease if you haven't established the gene-disease relationship yet. So we really start with gene curation, and then we can move on to variant curation using the ACMG AMP framework for how we curate variant disease relationships. This is our team at the Broad and Geisinger sites, working to create a lot of approaches and infrastructure and support our expert panels. There are two other core grants, as well as a number of smaller disease-focused grants now, but really we encompass over 1,840 investigators across 45 countries that are participating in our expert panels to really bring expertise around different gene and disease areas to ensure this data is curated appropriately. And we make well-founded claims of gene disease causality and variant pathogenicity. And I thank NHGRI's funding for this program as well. So you can go to our clinicalgenome.org website and see the progress across many different expert panels, just a screenshot of some of them here from 42 different gene curation expert panels. And to date, we've curated over 1,583 gene disease relationships, which highlights that about 27% right now of these claims that have been published don't have sufficient evidence. So don't believe everything you read in the even peer-reviewed literature. Often some of these are still in the candidate state. And in fact, many of these limited and below never move on, having now looked over time. So you really have to do this kind of curation. We have also launched a more global collaboration around gene disease curation. called the GenCC, which stands for the Gene Curation Coalition. And we started this group with a number of partners that were supporting public resources for gene disease validity, like OMIM and the Gene to Phenotype database by TGMI and the Genomics England Panel app and Orphanet, et cetera. And we really wanted to align these public resources around gene disease validity claims. And so we did a Delphi survey to develop consistent terminology. You'll see that's very similar to the ClinGen framework that all the groups have either adopted specifically or mapped their terms to. And now that allowed us to be able to share all of our claims into a single common database. Now, in supporting the GenCC, we also discovered there were many clinical labs that in developing gene test panels were doing this sort of curation to define which genes should go in their panels. So we brought many of the clinical labs and now other platforms into this fold. We now have 12 different groups. Our first publication is on medRxiv on this project, but all of the data is accessible on the GenCC website. I like to call it the Clinvar for genes. Like Clinvar, where you submit claims of variant pathogenicity, here anyone can submit claims of gene disease validity. So over the 12 submitters currently participating, they're all listed on the right side here. We have over 15,000 claims of gene disease validity across over 4,500 unique genes. And this is just a screenshot looking at one gene where you can see different groups submitting different claims on different diseases. All that data is structured and organized so you can quickly scan and see what the state of any gene is. It's also all downloadable for you to use in your own pipelines. Now, how is this curated data from either ClinGen or many other groups being used? So this is just a screenshot from Invitae's website showing that when the experts in Brugada syndrome published their paper around actually disputing 19 of the 20 genes that were part of that, Invitae changed its panel and it now has only one gene. Investigators can manually add on preliminary evidence genes, but the core panel is just one gene based on that curation. We also, this is just a screenshot of a report from one of our rare genomes project patients getting results back. We use the ClinGen framework to score those genes that have been newly discovered to make sure it's transparent, the level of evidence. And sometimes those reports are inconclusive because there's not enough gene disease validity. And we will state that, there still needs to, the association is not conclusive yet, to define that gene disease relationship. But a lot of the times these families then engage and find other families around the world. And so we partner with our families to help build the evidence when we haven't been able to come up with it on our own. So that's a bit about gene disease validity and creating global resources that we can all work together to build the necessary evidence. Of course, variants are a larger beast to tackle. There isn't a finite number of variants that will exist in our genomes. So we have to take a broader approach in ClinGen for tackling this. There will be not enough expert panels and experts to classify all the variants. So instead, we take a four-pronged approach. We develop common standards and distribute those standards, both through the ACMG AMP framework that we're working on updating, as well as ongoing guidance from the ClinGen Sequence Variant Interpretation Workgroup, both co-chaired by Leslie Sicker and Stephen Harrison. We also support and work closely with NCBI that maintains the Clinvar database to enable public sharing of variant classifications. This creates transparency and crowdsources the work. And over 1.2 million classified variants from submitters all around the world have been sharing those classifications. We also work between labs. Because we all submit to Clinvar, we can see when we're differing in our classifications, and we've been working to resolve those differences between laboratories. Now, when the labs can't come to agreement, we then push those on to the experts. That's where these ClinGen expert panels in variant classification come in. They have to go through rigorous steps to develop their rules and validate them. They now have FDA recognition for the rigorous process we go through, but those classifications also then get submitted to Clinvar. But it's a lower throughput pipeline because of the rigor. So we will still need everybody sharing their classifications from their own laboratories and sharing common approaches to this. I think the best place for us to all share that is still Clinvar, but you can see 40% of the claims in Clinvar are still variants of uncertain significance, this 40% of the pie. And that's just for the 1.2 million so far. We know there's over 760 million variants in Nomad, and there will be more coming. So to date, most of the data is coming from clinical laboratories in the US. If we get rid of the US on this graph and look a little more deeply, there are submissions coming from many other countries, but we still need more. Those of you here in the UK, shown here, in China, others that I know are on this call, let's all step it up and really share this data. There's much more to come in. That said, for those of you who use Clinvar, it's very useful and powerful, but generating knowledge is labor intensive. Reading the papers, contacting clinical labs to request patient data that's not directly in Clinvar. If we're going to scale this and really tackle, all the variants in the genome, we need to have better interoperable primary data sources that we're working from, as opposed to looking at pedigrees in picture form and reading evidence summaries in Clinvar. So how can we make the primary data more accessible and scalable to do this work? So one is population data, better allele frequencies. There's been work over the years to share aggregate allele frequencies from these data sets. The largest data is available from our Nomad database, currently at 183,000. We are currently working on the V4 release that will start with over 850,000 exomes. We're QCing and removing related individuals. We anticipate launching over 500,000 exomes and genomes in our V4 relaunch coming soon. And then we'll turn and that will include the UK Biobank data plus hundreds of other cohorts. And then once we launch that, we'll turn our focus to large genome data sets, particularly the All of Us genomes. We've already generated over 185,000 genomes to date from that program. But one key thing I'll point out is Nomad is diverse. You can see we've gotten more diverse over time, but it's not diverse enough. And in fact, it still doesn't have enough data, even when we launch the 500,000 in the next release. What we need is enough data to reach saturation of variation. And this is a figure from recent Nomad publication showing that we are starting to approach saturation for CPG sites, but most sites of sites in the genome, we are not saturating the opportunity for variation to be present. And if we saturate, we can actually use the absence in Nomad as evidence. Right now, absence in Nomad doesn't mean enough. You just need to be consistent with your pathogenicity evidence. We need to saturate, and then we can actually use this as the absence as evidence. I mentioned earlier that sharing data globally can be challenging in other countries. And so we are now exploring Nomad Federation, where other groups could do the same QC and joint calling and other approaches that we take, apply them to their data sets, and then we can aggregate the summary statistics together and represent them on a single website. And we're working right now with Daniel MacArthur, and I've had conversations with Nikki Mulder about doing that with Aboriginal data from Australia and more data from Africa. These are some of the most diverse data sets, and they will be critical in informing genome interpretation. And I'd like to see lots of this effort around the globe, and we're happy to work with others who can adopt the standards for the Nomad workflow and be able to allow us to bring more data together. Do want to acknowledge a lot of talented people working on the Nomad database, both our council, shown here at the top, it's leading this effort, as well as some of the core members of the team and the Broad Genomics Platform, Data Sciences Platform, Taukowski and Mutha Labs, who are doing structural variants, mitochondrial variants, all contributing to this platform that's been critical for genome interpretation. However, many of you use the Nomad database, sometimes go in there and you find your variant in one person, you're like, gee, I really would like to know the phenotype of that one person in Nomad. And unfortunately, based on the agreements we have with data, we cannot share the phenotypes. That's where we need better individual level databases that are phenotyped and we have access to the phenotypes. UK Biobank has been a critical resource. The All of Us research program is now sharing lots of data, and soon, starting in March, you'll have access to widely shared genomic data. We'll have the first 90,000 genomes released in March. Already global efforts, sharing data across biobanks around the world through the GBMI effort that's already sharing data, and lots through the IHCCC, lots of biobanks being cataloged with commitments to share data. And we hope that data is coming online soon and over time. I will say that some of these global biobanks don't always have good representation of rare disease cases. And so I do highlight up here in the corner some of the more rare disease focused databases that will be critical. The Decipher database in the UK has been an outstanding resource for rare disease phenotyped cases that have been publicly shared, over 30,000 cases. Other databases like Variant Matcher and Geno2MP in the US. Now I mentioned the CMG data in the Anvil platform. So we're starting to get better phenotype, both rare disease and general population biobanks shareable. That will be critical to really inform much of the variation and its pathogenicity. Also, more tools in terms of computational predictions. These programs haven't been perfect, but they're getting better. Debbie Marks's lab launching the recent EVE platform, the PriMed database from Kyle Farr's group, and most importantly, scalable functional platforms. And there's an organization called the Atlas of Variant Effects, bringing communities together that are working in these MAVE platforms, multiplex assays of variant effect, some on the MAVE-DB database. This is just a screenshot from saturating mutagenesis experiments across large stretches of genes that are really powerful to informing the effects of variation that will be critical to scale our knowledge and our predictability of what the effects of variation will be. So I agree with everybody. We still will have a long way to go. VUS term isn't becoming obsolete, but I think we can make major progress in this 8-year period. Our last one that I'll tackle is the regular use of genomic information will be mainstream in all clinical genetic settings. And here I'm going to turn to the hat I wear on the Chief Genomics Officer at MGH. But first, let's get your own opinions. Will the regular use of genomic information be mainstream in all clinical settings, not just medical genetics, but all clinical settings by 2030? Yes or no? Well, I'm happy to see more of you are confident in our advances here. Perhaps the innovation happening in the UK is helping with that. So I really am delighted to see this. So what are we working on in my hospital environment? So I formed this genomic medicine unit, really helping with the integration of genomics into the practice of medicine. This is sometimes more challenging in the US, where we operate much more siloed and very complex lack of national healthcare system. And so just working, we created an implementation team, which has physician representatives of every specialty across the hospital, working with them to define pain points in operationalizing genomic medicine, issues of lack of genetic support, particularly for low volume genetic testing clinics or primary care, a high volume of asymptomatic patients with genetic risk and where those patients go because they can't go to specialty clinics that are focused on symptomatic patients, referral and triage challenges, inability to understand our genetic utilization because it's not a national healthcare system, inconsistency getting consent and lack of support for genetic test ordering, just basic things that we struggle with. So we've been trying to tackle these. We've created a model for floating genetic counseling services, splitting genetic counselors across different clinics to help support their needs. We launched a common consent form that was down to 2 pages, translated into 6 languages and across the 20 different hospitals within the Mass General Brigham system. We launched a landing page for all the genetics clinics across the hospital and staffing a common phone number and email address to help with referrals and triage, all infrastructure. We also launched a new preventive genomics clinic to tackle the increasing large size of the patient population that is at risk, but not yet symptomatic, trying to target genetics at an earlier stage in the development of disease. And so we've been supporting this preventive genomics clinic with individuals that have family history, et cetera. Also launching an e-consult service where, and this is particularly targeted to primary care, where a patient might come in with a chief complaint of my brother or my sister died of breast cancer, my mother died of breast cancer, I'm concerned, what should I do? And those primary care physicians aren't often quite prepared, and they can send a query to our team, and we can give them guidance electronically and allow them to learn and offer these services themselves. And then lastly, we recently published the results of a randomized clinical trial comparing genome sequencing to standard of care across many different clinics in our healthcare system, showing that genome sequencing could not only make the same diagnoses, but catch more diagnoses in those individuals, really showing the promise of genomic medicine as opposed to standard of care. So with that, I'm going to finish so we have time for lots of questions. I will say there was one thing that they didn't add to this bold predictions down here at the bottom, that this darn COVID pandemic will end. So I don't have a poll for you to all weigh in on whether in 8 years this thing will end, but I sure hope so, because I've spent too much of my time involved in offering COVID testing. We're now at over 30 million tests since we launched this test. We had a little lull in the summer when we all thought this was coming to an end, but it is still strong. Although I will say on the positivity rate, we're having some great progress. Many other countries are as well. So hopefully we're coming out of some of the intensity of this. But with that, I do want to thank, I tried to highlight many of the teams I'm working with and collaborators around the world. This is some of the core people in my group. I get to stand up here and talk about all the great work, but it's really all of these people who do this work day in and day out that make all this stuff happen, as well as just an amazing set of collaborators around the world. And I'm happy to stop there and take questions. Thank you very much for your attention.
Heidi, brilliant. Thank you so much for a really fantastic tour de force. My name's Annika Lukeson, and I am the director of the Centre for Personalised Medicine. I've had the great pleasure of chairing this Q&A session. So could I ask people, I see people already started putting questions in the Q&A session. I've got some passed on to me as well. So I've got more questions than we can possibly deal with, I think. But if I could just start while we're gathering some of those and refer back, and ask you a very sort of general question, referring back to some of your poll questions. Mm-hmm. So I think you've shown beautifully how your global approach has really helped understand the biological function of human genes. And I wonder how much you think this approach can be used or adapted to look at how these gene functions might vary, say, across tissues or across time, or only be a risk factor on a certain genetic background, as you refer to sort of diversity issues. And I suppose what I'm referring to is that I think the discourse about genetics is still very often it's a causal gene or not, and how do we tackle that bit that's a bit more grey, where it's an important part, but dependent on so many other factors.
Yeah, we tackle this from the rare disease vantage point often because it's simpler. These are larger effect mutations in these genes that have dramatic effects. But if we want to tackle common disease, these are often much more subtle effects. They're often non-coding, and they may be in regulatory elements that only control the genes expressed in a certain tissue, and that's why they're only contributing to coronary artery disease or diabetes, certain things. And so I do strongly believe that tackling the non-coding portion of the genome that is often controlling the temporal and tissue-specific regulation of genes and what they do, and really embracing the fact that a gene is not a single gene. It creates many transcripts that have different structures, and starting to understand what an isoform does in this tissue or this time will be important. So really more, GTEx has been an amazing resource, but it's been largely a single point, mostly adult tissues. And we need to have more temporal analyses, tissues from newborns, tissues from early development. Some of that might have to be done in vitro, where we use other methods to stimulate development of tissues and cells and look at them in the course of manipulating expression and trying to mimic developmental stages and doing single cell analyses that we can really tease apart what's happening in different cell types with different triggers and of different regulatory pathways. So all of these tools and approaches and data resources are going to need to be developed, and different groups are going to contribute different things, but by sharing those data sets and those resources where we've each been manipulating different parts of the system, we can take advantage of that knowledge to inform what these genes are doing and how they're contributing and what their functions are in human.
Yeah, brilliant, thank you. Another question is, I mean, I guess your answer might be both, but I think it's still an interesting question. Which would be more immediately powerful in answering some of these questions? Collecting more data on larger populations of patients, or getting better at sharing and analysing the data that we already have in the system?
Right. It's a good question. And it's hard to say that one is more important than the other, because in a lot of ways, you don't want to, try to define right now all the things we need and the format we need them in and try to collect all the data and then worry about sharing it later, because it is through the process of sharing and working with those data sets that we realize we didn't ask the right questions, or we didn't standardize the data in the right ways, or we thought we did, but then when we try to combine it, we realize you interpreted this standard differently than I interpreted it, and now our data doesn't work together. So I love a quote that Eric Lander said when we were launching the Global Alliance. He said, "Rough consensus running code." And what that sort of meant was, let's come together with rough consensus about standards, but just start running the code, running the analyses, sharing data. We'll figure out where things aren't working, and then we can iterate on it. And it's just going to need to be a constant feedback loop where new technologies and approaches are coming that need new standards. We'll start figuring out how to use them. And so I would argue that sharing early is in fact equally as critical as starting to amass the data sets and realizing where we haven't collected enough information or detailed enough data.
And do you think we're beginning to address the diversity issue that way? Are we heading in the right direction?
We at least recognize the problems. That's problem number one, and recognizing that we need to collect diverse data. We are tackling that in the All of Us research program. For example, 50% of the samples collected are considered underrepresented in biomedical research. 50% is representing underrepresented races, and nearly 80% underrepresented in biomedical research, which also includes demographics and gender and other socioeconomic factors, age,
Fantastic.
all reasons that we are underrepresented. And we need, recently I've seen differences in associations because of looking at healthcare data. And what I would argue is where an association might not be seen in Blacks where it was seen in whites. Well, that could be because the Black population hasn't had equivalent access to healthcare sufficiently to actually document those phenotypes in the electronic health record. And so our association studies that are relying on healthcare gathered data are not showing the associations because of disparities in access to health care. So there's important. So many different aspects of the system we need to fix. It's not just going and getting the samples, it's also making sure we phenotype effectively these individuals who haven't had equitable access to healthcare. So there's, every time we look at a problem, we realize there's actually more problems
behind it. Yeah. So you spoke about the constant innovation to support genome interpretation. Is there a development on the horizon that you're most excited about?
Yeah, so that's a great question. So I do think that, some of the advances in multiplexed assays of functional assessment are really exciting. The ability, we just can't, find a single variant in a patient, and then go out and do a functional assay one at a time, and then try to bring that result back in the timeline a patient needs it. And so some of these, MAVE sort of platforms, multiplex assays of variant effect, I think are some of the really exciting things that will allow us to actually utilize that data in real time when we find the variant in an individual. That said, trying to develop assays that represent the true biological function of a given protein. Some of these proteins we don't have assays for. And so it's very difficult to do these scalable multiplexed approaches. So I think I'm really excited about some of these platforms and approaches, but we still need to really innovate in having an assay for every gene and its many functions.
Yeah, brilliant. Combining several questions here, I think. So what do you think are the main barriers to open data sharing? And I guess in that I include phenotype sharing, because that's probably your biggest barrier, isn't it? And have recent legislation changes, such as the European General Data Protection Regulation, had an effect already that you can see, or a good effect or a bad effect? Can you comment on that?
This is challenging. And I think the concerns over the privacy and security of data are both one of the most important things we have to pay attention to, but it's also been one of the biggest barriers to data sharing. And when we're sharing, genomic data and highly detailed phenotypes, these are in essence can be identifiable. That said, you can only identify a person by their genome if you already have access to their genome. So, so there's both the hypothetical question, but then there's the practical question. Of course, with people sharing ancestry information online, there is real notion of being able to identify an individual by their genomic data because of the way that people are engaging in data sharing. So I think the way that we're going to have to tackle this, and this is one of the approaches that the Global Alliance has taken, is to engage the participants, to say, let's not just operate in our box over here and try and create a system that is perfectly secure and private and promise everyone that there's no chance their data will be, breached. But in fact, engage individuals and help them understand the benefits of data sharing and how they can benefit and how they can contribute to science and realize the benefits of science. And then they will be more readily apt to take some risk, because we all have to take some risk when we think about data sharing, but say that in balance, the benefits outweigh the risk, but make sure that we're engaging the community as partners in those decision makings, and that we're making these decisions as a global community. And I think in those regards, we will be able to tackle some of these challenges in data sharing and make advances.
That's such an important point. Thank you. I see the time is coming up to 1 o'clock, and we did promise that we'd finish promptly. So I want to say thank you so much for presenting a fantastic talk and fantastic Q&A session as well, and particularly getting up. I understand it's not too early for you, but for everybody else, it's 7 o'clock in the morning for you right now, so that this can go across multiple time zones. And I hope that our colleagues in Hong Kong, Macau, and China have managed to enjoy this in their evening whilst they're celebrating the Chinese New Year. So Happy New Year to our Chinese
Happy New Year.
Colleagues.